Thank you. Thank you so much for the kind introduction. And yeah, I'm standing here today because of Dr. Leonard, and I'm so glad things worked out. And I'm happy to be connected to pediatrics as well, although I'm housed in epidemiology. I'm always looking for trainees and fellows, and anyone that wants to work on research with me, feel free to reach out. So today I'll be talking about environmental epigenetics, and perhaps this might be a new concept to you. Some of you might have heard of epigenetics and how this applies to children's health. And I'll go over some of my research as well. So what I'm hoping to accomplish today is to really define epigenetics for you. So we all leave the room with a working understanding of what epigenetics is and what epigenetics is not. I'll try to motivate this in the field of pediatric research, thinking about fetal epigenetic programming. And this is one of the concepts that really got me interested in the field. I'll give you a couple of examples, one related to prenatal smoking and how that might program placental methylation and how it's related to things like birth weight that we care about. The other one is related to more respiratory and allergic disease, looking at the nasal methylome. So we're really excited about this new tissue that, of course, a lot of people have been thinking about nasal samples since COVID, but something novel that we have been collecting as well, and how it relates to respiratory and allergic disease. And finally, I'll talk about aging, and you might be wondering, why are you talking about aging in a pediatrics ground rounds? But I think we've been trying to frame that aging is really starts taking a conception, right? So once you are conceived and born, that aging trajectory is set very early on, and this is something that we're really interested in. My work encompasses epidemiology. We also use epigenomic tools as well as big data and machine learning tools to look at how the environment affects our health and disease throughout the life course. And then I'll end with some take-home points that I hope is emphasized throughout the presentation. So what is epigenetics? So epi comes from the Greek, literally means above or upon genetics. So these are changes that are on top of the genetic code. These changes are changes in gene expressions that are not dependent on the genetic code. So you're altering the expression of genes without altering the genetic sequence, and this is key. These changes are also stable. So this is what makes it very attractive as a biomarker compared to, say, transcription or RNA, that RNA is less stable. So these changes are fairly stable once they happen. They might also persist mitotically. And when I say persist, they persist from one cell division to another. There's some debate. If you talk to individuals doing research with nematodes like worms and plants, there's pretty clear evidence that go from one generation to another. But in mammals and humans, that's debated. So there's quite a bit of debate there. And that actually goes against the idea of evolution, that you can pass down information from one generation to another through epigenetic marks. So still highly debatable. There's not enough data or evidence just yet. So if you take anything from today's talk, I want you to know that epigenetic marks are tissue and cell type specific. So if you think about all the cells in your body, right? So your brain cells or your heart cells, they look very different and they have very different functions, yet they have exactly the same genome for all practical purposes, right? So the DNA code is the same. So how are two cells that do and act very differently achieve this? And one process is through epigenetic programming. So very early on, once the zygote is formed and there's pluripotent stem cells and some of the stem cells that are going to become different types of cells and tissues, there's a process of epigenetic reprogramming that happens to form the different layers like the ectoderm, the mesoderm, the endoderm, and even the germ cell layers that are, these cells commit to become this type of cells and commit to being this type of tissue very early on. And this is through epigenetic programming. So you go from having the same genome, even within the same organism, as to having different cell functions and different cell types through epigenetic programming. So epigenetics contributes to tissue differentiation and cell type fate differentiation as well. So hypothetically, each cell type, within each cell type, there's a unique epigenetic signature, which, as you can imagine, is a huge challenge for us doing research, and also holds a lot of promise as well. So the NIH has realized the promises of the epigenome, and they have invested heavily in trying to characterize the epigenome. This is very similar to like the Human Genome Project, to try to code the entire human genome. The epigenome is a little more tricky. Right now, we're up to about 111 human epigenomes, whether those are cells or tissue types. It varies a little bit once we start digging into cell types, right? As some of you have taken immunology, I'm sure there's multiple cell types within each cell type. There's sub-cell types as well. But what I want you to know, relevant to us, is that there's also the fetal epigenome and the child epigenome. And they differentiate between adult and fetal tissue. And the reason being, although this can be the same individual, their epigenomes are going to be pretty different, even for, say, a simple or one structure like the brain. The epigenome of adults or the same individual is going to look different than the epigenome of the brain of the child of the same individual when they were a fetus or they were a child. So this drives the point home that there's a component of time. So the passage of time gets recorded through the epigenome. Of course, it has to do with development and maturation as well. But that holds a lot of promise to me in terms of trying to optimize health outcomes very early on, identifying biomarkers as well. But at least now we have some sort of what a healthy epigenome might look like. We're not there yet. We don't have like gold standard references, but we're beginning to understand what normal and abnormal looks like for epigenomes. I'm going to talk mainly about methylation of DNA due to time, and this is what I think is ready for prime time, and there's some clinical applications to methylation measurements of DNA. These other measurements like histone modifications and non-coding RNAs are also considered epigenetic modifications, but we're not just there yet to try to translate this, and even for population studies. So when I'm talking about methylation of DNA, I'll be mainly referring to methylation of cytosine molecules. So this is one of your nucleotides, one of the base pairs of DNA, the cytosine, can become methylated at the 5' position of this ring by the addition of this small methyl group to it. And then once you look at it, it's still a cytosine. It remains as a cytosine, but it now has this tiny methyl group attached to this position here in the ring. And that's an epigenetic modification. Histones are the protein complexes where DNA can actually wrap around and you can block transcription. For example, if the DNA is tightly packed, as you see here, the target gene is not transcribed. And then non-coding RNAs can interfere with transcription and translation in several ways. But for example, microRNAs can bind to either stabilize or destabilize translation of the protein. So these are 3 examples of epigenetic modifications. I would say the most widely studied is methylation of DNA, that we can actually study it at a single base resolution in the genome. And I'll show you how we do that. So why methylation of DNA? Why do we care about DNA methylation? So DNA methylation is thought to be a molecular switch. So then again, here you have your cytosine. It undergoes methylation by the addition of this methyl group. And here we have SAMI, which is one of the methyl donors, can donate this methyl group. And there's a specific set of enzymes, for example, DNA methyltransferases, that can actually steal that methyl group from SAMI and donate it to the cytosine. So that's really important for epigenomic maintenance of our genomes. So one of the first realizations in the field is that these CG dinucleotides, so a cytosine followed by a guanine, don't occur at random. So there's these clusters, and oftentimes they occur upstream of genes. So here you have your gene, your favorite gene, and you have what I'm calling a CPG island, which are CG dense regions. So these are CG dinucleotides that are pretty dense upstream of the genes. And if this CPG island is not methylated, represented here by these lollipops, so these are just regular cytosines followed by guanines, gene expression can occur. And then if it's heavily methylated, gene expression is repressed. So now you have a really elegant switch. And this is not an all or nothing event. This is more of a volume that you can actually regulate expression to a certain extent. So remember, this is not a binary outcome. We're looking at degrees of methylation, so you can methylate some cells and demethylate some other cells at a specific genes, and then gene expression might be regulated that way. So the whole premise here is that we now have several examples where methylation differs between disease and healthy state, whether it's tissue or conditions. So the whole hypothesis is that methylation of DNA might be responsive to environmental conditions because it's malleable and might be responsible for health and disease risk throughout the life course. So this is one of the reasons I became so interested in methylation of DNA. So there's this process of fetal epigenetic reprogramming that happens early during fetal development. So here on the y-axis, you have the degree of methylation. And then on the x-axis, you have the process of development. So very early on, the sperm and the egg have some degree of methylation. Upon formation of the zygote and fertilization, what happens is that methylation gets erased for the most part out of the entire genome. So it's kind of resetting the program. You're erasing all the methylation marks across the chromosomes, with an exception of a few imprinted genes. And I'm sure most of you know about imprinted disorders, right, where there's methylation either on the paternal or the maternal side of the genome. And if there's aberrant methylation, maybe the two genes are heavily methylated and therefore silenced, or maybe they're not methylated, disease may occur. But this is the exception. For the most part, the entire genome gets demethylated, and then there's the remethylation event where the cells are being programmed to become specific cells, and they go to form somatic tissues and so on. So to me, this is a very critical window of development, right? This is where there's a high demand for metal donors like folate. This is why one of the reasons why folate is important during fetal development. But because I'm in public health, I'm really interested in prevention at the population level. And particularly when we study chemicals, we think about setting standards for regulation. And I think we should be looking at this because hypothetically here, we might be able to protect 3 generations if we set standards based on this critical window. We have the F0 generation, the F1 and the F2, the germ cells as well, that we can protect very early on. So this is key in terms of epigenetic programming, and we study a lot of exposures during pregnancy and then follow children throughout life in a few cohorts to try to understand what are the implications of exposures, what are the implications to child health and development later in life. So I wanted to present a little bit of an older study, but I think this study is an experimental study that really shows nicely what might be happening here in terms of epigenetic programming. So this is the agouti mouse model. There's this regular variation in coat color, but most of the agouti mouse are this pseudo-agouti color or heavily mottled mice, so more of a brown color. Once in a while from a litter, you get this yellow agouti mouse. There's natural variation, but in this experiment, mice that were exposed in utero, so this is a litter that was exposed in utero, with increase in BPA, you get a higher frequency of offspring that had this slightly mottled or yellow phenotype. And BPA is a plasticizer that was put in bottles. Now it has been phased out mostly and replaced by BPS. And BPS has a shorter half-life in the body, so we excrete it quicker, but yet there's still endocrine action to it. That's a side story, but you probably have seen a lot of the plastic bottles that say BPA-free for multiple reasons. But here, this is an early paper, 2007, showing effects at a specific phenotype like coat color that is so visible. What they were able to show in here is that the agouti gene was actually demethylated among exposed offspring, that moms that were exposed to BPA in their diet, compared to animals that were in the control diet where there was no BPA. And why does it matter? Well, of course, coat color is very visible, but this more rare yellow or slightly mottled agouti mouse tends to resemble insulin-resistant phenotype. So basically, they're more prone to obesity and to develop insulin resistance compared to the pseudo-agouti mouse that is more frequent in the population. So here you have a prenatal exposure, which is BPA, a plasticizer, a known endocrine disruptor, altering methylation marker to a specific locus that controls coat color, that leads to potentially a phenotype that might resemble insulin resistance and diabetes. So to me, this is a pretty good example of how a prenatal exposure might program the offspring health and development. So how do we do this in human studies? So we know that we're exposed to multiple chemicals and stressors, where there are social chemical stressors. That leads to an internal dose, for example, lead. We know that lead crosses the placenta. It also crosses the blood-brain barrier as well. So the question becomes, what we try to do in our work is to try to answer the question whether that influences the epigenome. And as I mentioned, there's multiple epigenomes, so that becomes a big question. And then whether these epigenetic modifications, okay, well, methylation changes, does it matter? Does it affect newborn health? And things like birth weight and gestational age. But why do we care about those things? Is because we are really thinking about life course health, disease risk from an early life exposure. I am missing a very important piece here, and I personally feel responsible for kind of contributing to more of the maternal health research, but yet we don't include parents, right, or the paternal side. So the reason being is because it's fairly difficult to catch couples prior to conception, although there's a few studies that are trying to do this. I work with a few colleagues that are trying to look into IVF cohorts, for example. So these are couples preconceptions. So there's now mounting evidence that also the paternal epigenome, I just told you everything gets erased and reestablished. That was a very broad stroke of saying epigenetic reprogramming. There's some evidence that some genes, including some of the imprinted genes, might be passed on to the offspring later down the line. So it's not that, you know, the lack of data is perhaps like design, because we have mostly designed cohort studies follow pregnant people and not the contribution of the paternal line. Okay, so how do we do this in humans, right? So in the example of the agouti mouse, they looked at the agouti gene. That makes sense, right? That gene was known to control color. But how do we do this in humans? You know, we go gene by gene, and that's actually what people did very early on, very similar to genetics. For example, people kept studying P53 because P53 controls cell cycle and other things, but that becomes an unscalable thing, right? Where you're testing gene by gene, and there are thousands of genes, and each gene might be mapped to hundreds, if not thousands, of individual cytosines. So that's what we did very early on. But luckily enough, biotechnology caught up and sequencing technologies as well. One tool that we have now is these high-density microarrays, very similar to genotyping the arrays. So if some of you have done some of the genotyping commercial available kits, this is basically what they're doing. These microarrays are able to survey the genome, not entirely, but pretty well for CPGs. We're getting close to almost 1 million CPG sites. The latest iteration of this microarray is getting close to 950,000 individual CPG sites. And it has pretty good coverage. It covers a lot of the genes in the RepSeq genome. And it does it at a single nucleotide resolution. So, you know, for this specific CPG, what is the degree of methylation and where in the genome is that located? So you actually get location. We performed these hypothesis-free epigenome-wide association studies, very similar to GWAS, where there's genome-wide association studies for SNPs, what we call these EWAS, which are epigenome-wide association studies. The data is very high dimensional, right? So for every single individual, we get about a million data points. So a number of parameters is huge, and we run into bioinformatics and methodological issues. So there's a lot of work on that side into the methodology that I won't be going over today. So I wanted to give you an example as it relates to child health. And here we looked at prenatal smoking and placental methylation. So I'm on purpose not trying to show you some of the blood findings. Very early on, people were studying methylomes of leukocytes because everyone collected leukocytes for DNA sequencing or DNA genotyping. So that was really available. But then with epigenetics, we realized that tissue matters a lot. So, right, so we had to design new studies to collect novel tissues, and in this case, the human placenta. So I don't have to convince you that studying smoking is very important clinically and also for public health. So smoking causes low birth weight. It also reduces fetal growth. In the US, there's quite a bit of variation. The prevalence is about 5% of pregnant people reported smoking during pregnancy, but there's quite a bit of status variation, as high as 25% in West Virginia, and California is doing pretty good. We're getting close to only 1% of pregnant people reported smoking during pregnancy. So why do we study the placenta? Well, the placenta is the master regulator of the fetal environment. It allows for waste and nutrient exchange. It also has endocrine functions, so it produces hormones, communicates with the maternal and fetal side. So in this study, this is what a prospective birth cohort called Gen3G from Sherbrooke, Canada. We recruited 441 mother-child pairs. They self-reported smoking during pregnancy, and we collected placentas at birth. We queried the epigenome of the placenta using these microarrays that interrogate over 700,000 individual CPG sites. The cohort was generally healthy. The only thing that I wanted to highlight here is that only 38 moms reported smoking during pregnancy. That's a little bit higher than the prevalence in the US, I guess, the average prevalence, but 8.6%. And you'll see that, you know, although the sample size is relatively small for some of the genomic analysis that we do, we were still having power enough to, statistical power to be able to detect associations. So this is the framework that we were working under. First, we did confirm that pregnant people that smoked during pregnancy had babies that were on average 175 grams smaller, newborns, compared to non-smoking pregnant people. The question that we wanted to ask is whether smoking influences methylation of the placenta, right? So those methyl marks, whether they're altered by smoking prenatally, whether those methylation marks actually predict birth weight, are they related to birth weight? And then the real question that we're trying to ask is whether the amount of the effect out of 175 grams, that's an estimate, how much is it going through methylation of the placenta? Is there any evidence that some of the effect is actually mediated through placental methylation? So we perform a statistical mediation analysis. We perform an EWAS here, an EWAS here, and then finally we perform a statistical mediation analysis to look for evidence or not of statistical mediation. So I wanted to show you the impact of smoking on the placental epigenome. This was one of the first few studies to look at this question. It has been looked at blood and leukocyte cord blood before. What we were able to document is that the smoking effect on the epigenome of the placenta is widespread across pretty much every single chromosome. This is a Manhattan plot. If you're not familiar with this, this is the negative log of the p-value. Every single data point is an individual CpG site, and this is mapped to the 22 chromosomes. We excluded the sex chromosomes. So what we see here is that every single chromosome in the placenta is being affected by smoking. This is a volcano plot. Here on the x-axis, you have the effect size, and then on the y-axis, you have the negative log of the p-value. So these are very significant results, 10 to a negative 30. And then a 30% decrease in methylation for smokers. The other point that I wanted to make is that, for example, this LAKR1 gene that is differentially methylated, the signal is so strong that you're actually able to classify people that smoke and that didn't smoke during pregnancy just by looking at the methylation marks with pretty high accuracy. And I'll show you some AUC curves that I think I included here as well. So the effect sizes are strong and they're widespread throughout the epigenome of the placenta. This is somewhat of a busy table. This is the CpG ID, the chromosome, the position within the genome, and the specific gene. This is the effect size that is direct. So this is the direct effect size of smoking during pregnancy on birth weight. And this is the mediated effect. So this is the pathway, the indirect pathway that we're trying to characterize. We found pretty strong evidence among 7 CpG sites that map to different genes, including some of the CYP. I'm sure you have heard of some of the CYP genes that are metabolized exogenous exposures. But particularly what caught my eye was this PVX1 gene that had pretty strong effect size, 168 grams, that was mediated through that specific pathway, going from smoking to lower birth weight, specifically through the methylation of the PVX1 gene. And then some of the effect sizes were a little smaller, like 88 grams. We did test for exposure mediator interaction, so this is more of a technicality, but we proposed that the level of methylation that you start with, so how much methylation the placenta had to begin with, or at conception or formation, influences whether smoking has an effect on birth weight or not. That's what this interaction test is telling you, and we found some evidence of that. But the effect sizes were quite strong. I won't go too much into detail, but we did find that the placenta was very sensitive, as you saw, across all the chromosomes to maternal smoking. We found evidence of mediation with a specific set of genes. This PVX1 gene, I didn't know that it existed, of course, before doing this, because this is untargeted. But after doing more of a literature search, PVX1 seems to regulate osteogenesis and skeletal pattern, rinds of bone formation. And we also found one other study where PVX1 in cord blood was associated with birth weight. So that was more evidence that this might be causative on that pathway. And then we proposed this exposure mediator interaction that was highly relevant for epigenetic epidemiology. And this has been done for cord blood. The signatures appear to be different. So this is highlighting the relevance of studying different tissues as well. So I want to briefly mention that the evidence is mounting now. Exactly what we're trying to do is trying to investigate that indirect pathway. Of course, all of these studies with maternal smoking is observational studies because you cannot do a randomized trial, of course. But no one argues that this is not causal. there's now more evidence. For example, we investigated birth weight, but people have done studies looking at schizophrenia, asthma, ADHD, mediated through either placental methylation and cord blood. You can see these are some of the cord blood studies. This is a review, and these are some of the other studies that have looked at cord blood or placenta and different biomarkers like cortisol or different outcomes like ADHD or schizophrenia as well. So there's mounting evidence that there might be a causal role of the methylation influencing health and disease trajectories. So these are the AUC curves that I promised to show you. So the signature is so strong, even in a small sample size, that you're able, this is cord blood. So using the methylation marks associated with smoking, you're able to classify smokers and non-smokers. Well, there's other biomarkers to measure smoking, so maybe this is not so exciting. But this is a proof of principle. The other thing that really struck me, this is a study that looked at the methylome in leukocytes, so blood drawn 30 years later. So these are adults in their 30s. And they asked the question, can we actually predict whether the mom smoked during pregnancy with an AUC of about 0.72. So this is looking when you're an adult, trying to predict whether your mom smoked during pregnancy. And they also adjusted for environmental tobacco smoke and also on their own personal risk of smoking as well. So I think this is a proof of principle. As the biomarkers keep getting better and our technology keeps getting better, I think we're going to be able to predict more exposures. Now we have biomarkers for alcohol consumption. We have biomarkers for environmental exposures. We've been trying to build some biomarkers for air pollution as well. So in the future, this is not ready for prime time, but I do think as the technology gets better and our studies continue to improve, we might be able to predict previous exposures all the way back to the prenatal period, but also early in life potentially. So I want to tell you about another tissue that we're very interested about, which is the nasal methylome. So these are nasal cells. We started collecting them before COVID, back in, I believe, 2016. Before, nasal collection was pretty standard. Well, it's pretty standard nowadays. So why do we study the nasal methylome? Well, it's in direct contact with the environment. So we're really interested in air pollution, and that's what we wanted to see, right? So a lot of the gas exchange comes through your nasal passage, and we're interested in air pollution, so it makes a lot of sense. Most studies have been studying leukocytes or blood, which of course is an inflammatory tissue, but it'll be more interesting to look at target tissue. It's also fairly accessible. If you wanted to collect respiratory epithelial cells, you have to go into the lung, which we have done in adults, but in children, you can imagine that probably parents wouldn't consent to that. So we did a study. Also, this is a prospective birth cohort in Boston, Massachusetts. The median age was about 13 years. We have about 500 participants, and we profiled their epigenomes with the microarray that I just mentioned, getting close to 900,000 individual sites. What I wanted to mention is that we sampled the nares. So we collected at the nares, and we did some piloting about collecting the inferior or superior, the turbinates, but the issue is, it's not pleasant to get sample at the turbinates, especially for children. So we selected the nares. You get less respiratory cells. You also get some squamous cells that are in the nose, but you get a pretty good proportion. About 50% of the samples should be respiratory cells that should reflect what's happening in the lung. So when we looked at different phenotypes, this is an enrichment of different pathways. So the signal was very strong for allergic asthma. So children with allergic asthma had multiple differentially methylated regions and genes. Increasing exhaled nitric oxide was also very similarly associated with the same pathways. And the signal for non-allergic asthma, just asthmatics, children that had asthma that did not have allergy, was slightly different. What was really interesting is that the neutrophil degranulation pathway was differentially methylated for allergic asthmatics, and also with increasing exhaled nitric oxide, which has been shown to be a causative factor in pulmonary disorders. Signaling by interleukins, particularly 4 and 13, was differentially methylated, so the entire pathway also showed up as a significant biological pathway, which is a central regulator of IgE and airway hyperresponsiveness. So that was also really interesting to be able to capture the disease process in the nose and the nasal methylome of these children. Now, I wanted to introduce the concept of epigenetic aging, and this is something that we've been studying in terms of biological aging in early life. So if you think about how do we keep track of age, right? When someone asks you how old you are, we report a chronological age, which is basically how many revolutions we have completed around the sun. But there's this common conception, right, that maybe, some people age at different rates, depending how much you exercise, your diet, how you manage stress, things like alcohol consumption and so on. So of course, the aging field has very, very interested in telomeres, and you probably have heard of telomeres. But if you tell me your telomere length, I wouldn't be able to tell you your age. We want to have, very accurate biomarkers if possible. So how do we keep track with a biological calendar? Well, telomeres are one way to do this, but now the field is moving to these omic-based clocks. And this is an epigenetic clock developed by Steve Horvath at UCLA. It's multi-tissue. So here's Leonardo da Vinci's Vitruvian Man, and you can see that he's highlighting that there's multiple clocks across multiple tissues potentially. What he did, he trained a model to try to predict chronological age based on epigenetics. This is a key plot. This is the methylation age predicted using machine learning. And then here on the y-axis, you have the chronological age. The error rate of this clock is about 3 years. So you're able to kind of predict someone's age with reasonably expectation throughout the life course, all the way from 0 to 100. Of course, when you zoom in into one of these windows, you see that there's deviations. So what we're really interested in is in those deviations. Why are some people biologically older? Why are people biologically younger? And I'll say a little bit more about this, but now this is being used for like crime scene investigation, right? It will be really useful to know the age perpetrator of a crime if you're able to get DNA. But there's other applications that perhaps are not as altruistic, and I'll talk about that. So we asked the question whether allergy and asthma had an influence on epigenetic aging, with the hypothesis that children will have accelerating aging. Our hypothesis was partially supported. So here we have children with asthma that also, this is an allergic asthma phenotype sensitized to IgE. Children that had asthma that had allergic asthma were on average about 1.3 years older compared to children with not asthmatic children. Total IgE was literally related to epigenetic aging, as well as environment-specific IgE sensitization. And also just not the allergic asthmatics, but the children with asthma, non-allergic, had also accelerating epigenetic aging, and also increasing exhaled nitric oxide was linearly associated with epigenetic aging. We hypothesized that better lung function, right, measured by FEV and FVC, if your lung function is better, maybe your biological age is younger. That was not supported by the data, but I think these children have pretty well managed asthma. They all have insurance, and pretty good insurance too. That was one of the requirements, or how we recruited for the study. So I think if you were to do this in adults, the story might look different, right? That better lung function hopefully is correlated with younger biological aging. Okay, so I want to conclude going over more epigenetic aging clock research. And I think this is, we've been able to convince, for example, the National Institute of Aging to give us a little bit of funding to study the determinants of these clocks early in life, with the hypothesis that some of these trajectories are set either prenatally or by early life events as children develop, and that these trajectories then set you into a course into health and disease. So the clock that I showed you was the Horvath pan-tissue clock. So this clock applies to multiple tissues, and it works across the life course. After this, now there's multiple clocks that have been developed, what we call second generation clocks. Some of them are more accurate, some of them only work in blood, some of them are only for specifically calibrated for children or vocal cells. So people have been developing with different goals in mind. There's 2 clocks that we've been working with, which are the gestational age clocks. So it's basically to try to predict gestational age of birth instead of chronological age. So to have a higher degree of accuracy, if you will. These are the night clock and the bowling clock. This is data from a prospective birth cohort in the Salinas Valley of California, the Chamacos birth cohort, in which we tried to predict chronological age based on the epigenome of cord blood. And you can see here that Boolean's clock is doing fairly well. The correlation is not as impressive as the adult clocks, but 0.67, that's pretty good, with a median absolute error of about 5 days. So that those deviations on average is about 5 days. So we have characterized the performance of these clocks. This is work performed by a student when I was at UC Berkeley, Saher Daredia. She was really interested in programming, fetal programming from maternal exposures of these clocks. So she looked at maternal triglycerides, lipids, preterm delivery parity, and infant sex. And with the more accurate clock, we got pretty consistent evidence that increasing triglycerides, lipids, preterm delivery and parity decrease the epigenetic aging. So this is a marker of immaturity, if you will. This is the way we're thinking about it. In adults, increase in epigenetic aging has been the risk factor. That's what people have been hypothesizing, and that's what's correlated with mortality and morbidity. In children, what we have seen now is that things that are known to be adverse for infant outcomes, things like increase in maternal triglycerides, lipids, and so on, are correlated with deaccelerated epigenetic aging as a marker of immaturity. And it's worth to note, I didn't go into details, but the acceleration is calculated as a residual from the gestational age of the chronological age. So this is completely independent of the actual chronological age. If you were to try to plot the acceleration measures, which are here on the x-axis against the actual chronological age, it will look like a cloud. There's no correlation by design. So the fact that preterm deliveries are showing increased or decreased epigenetic aging as a marker of maturity is reassuring for us, at least. One finding that really struck us is that out of all the social factors that we looked, income has an effect, and others have reported this, so maternal income has an effect on the infant epigenome. We have followed these children to age 7, 9, and 14, and we're now collecting more data at age 18 from the prenatal cohort. Children of mothers that work in the field during pregnancy had accelerated epigenetic aging pretty much uniformly across different clocks. And these clocks are designed to measure different things. They're all calibrated to measure chronological age, but some of them are more associated with morbidity and mortality. So it was pretty remarkable that at age 7, moms that's prenatal maternal field work was associated with increased epigenetic aging of these children pretty consistently across clocks. And I have not seen this for other exposures, even smoking, that I usually think about smoking as one of the exposures that is pretty harsh. We have not seen this. So there's something about this. We're looking into whether it's pesticide exposure, right? So these are farm workers that are working out in the field, whether it's, of course, the strenuous labor that they perform. So I think this is one of the social determinants or labor factors that might be associated with epigenetic aging of the next generation. So that was pretty interesting. So we have also studied ACEs. So of course, I don't have to tell you too much about ACEs. You probably administered these in your practice, but these are self-reported adverse childhood experiences that we collected, and we collected this from the moms. So by design, everyone that enrolled in the study had to be 18 years or older, and they reported on ACEs, and of course, ACEs are 0 to 18. So hypothetically, right, all of these ACEs are preconception, so before conception happened, reported by the mom, and then we looked at epigenetic aging of the children. Of course, this is a little bit controversial, as I mentioned, because this indicates the hypothesis that we're trying to test is that there's some acceleration of the clocks that is through generations. So it's from the mom, not prenatal, but preconception to the child after birth. So that's the hypothesis that we're trying to test, whether ACEs accelerate epigenetic aging of the child later in life. So this is the data that we have from the Chamacos cohort. Remember, this is the Salinas Valley. We did find evidence that ACEs, unfortunately, is quite high in the study cohort. About a quarter of expecting people reported having 3 or more ACEs, experienced 3 or more ACEs, and this was pretty consistent among the subsamples at age 7, 9, and 14, about 25%, the estimate throughout the 3 study visits. So this is work performed by a postdoc. He's now a faculty member at Emory, Dr. Jamaji Nawaji Emren, and he was really interested in testing the effect of ACEs on epigenetic aging later in life. What he was able to document is that indeed increase in ACEs was associated, or like 3 or more, 2 or more ACEs were associated with epigenetic aging of this multi-tissue clock that I mentioned earlier. So he was able to dig into the 10 domains of the ACEs to see which one it was that was accelerating epigenetic aging. And I thought, before when we met, I told him, I think it's probably the physical abuse or mental abuse. I think those are pretty traumatic events. And we were surprised to see that it was actually in our data, divorce was the one ACE domain that was driving this association. So parental divorce of the mom, right? Or not the mom, but as reported by the mom preconception. Another finding is that we found pretty consistent associations with accelerate or increase in telomere length. So this is estimates of telomere length at 7, 9, and 14 years of age. We thought it was, hypothetically, if you're accelerating aging, telomere length could be decreasing. But interestingly enough, increase in telomere length has been associated with cancer, which is one of the findings from the ACEs literature. And we're finding evidence here that the telomeres are of exposed individuals are longer compared to unexposed individuals. This is a little bit, we have to go back and forth with the reviewers. We have to pose multiple hypotheses. Maybe these are permanent socioeconomic conditions, although we try to adjust for things like education, SES, household support, and so on. So I think we adjusted for a lot of the social factors. So whether this is true transgenerational transmission is yet to be determined. What I wanted to end the talk a little bit about the misuse of some of these biomarkers. I just have a snippet here of a quote from a CEO of an insurance company. They're actually using the epigenetic clock to try to risk stratify people for insurance premiums. So he said, what we're trying to do is like precision medicine, but precision insurance instead. So basically, they're taking a buccal cell sample, estimating your epigenetic age, and risk stratifying you based on your epigenetic age. I have met providers and also people that are doing this. These are people, of course, insurance is not reimbursing people for this, but you can go online and order these kits and measure your epigenetic age. I'm not saying people should. I haven't done it myself. I don't know how much more information that will provide beyond, some of the information that we already have and what you would do about it. But my point is that I think we're running into a lot of ethical issues here. How this is used is very unclear whether GINA, for example, the law that protects against discrimination for genetic information, covers epigenetics. As I mentioned, things like smoking influence the epigenome. So people have tried to argue that epigenetics shouldn't be covered under GINA. Insurance companies are using it now to risk stratify people. There's a lot of the industry in health and wellness are also using some of these clocks to try to give patients back information as well. But I think it's dangerous, as I just showed you. There's both environmental and psychosocial factors that might influence epigenetic aging, and which are the populations that are going to be affected by some of these decisions of trying to use some of these biomarkers to either risk stratify or to provide or not provide different services. So I think it's somewhat of a dangerous path. I have proposed that we shouldn't be doing precision insurance. We should try to do precision environmental health, at least for what I do, to try to slow epigenetic clock ticking rates. I'm really interested in trying to design studies to test interventions. We actually have a study with a colleague from Florida. He's testing a parenting intervention. So this is an intensive parenting intervention with kids that are at risk with behavior problems to try to see if the parenting intervention actually reduces epigenetic ticking rates among at-risk youth. We want to reduce harmful exposure. So I want to try to understand what are the things that accelerate these clocks, and can we actually do something about it? And how can we optimize both the prenatal and the early life environment to really give children their best chance? So I think this is the way that we can use these novel biomarkers. I am a little bit concerned the way they're potentially being used and misused now as well. So just very briefly, I think in summary, what I wanted to convey today is that epigenetics is a very useful biomarker. I may be a little bit biased, but it provides a lot of insights into mechanisms, but it can also provide biomarkers as well. A lot of questions that I get sometimes is like, well, what about the other omics? Like we have done RNA-seq studies and so on, but the sweet spot of methylation or some of these epigenetic marks is that they're fairly stable. Once a condition established them, like for example, with smoking, they stay out there, and they stay out there with a sense that you can actually predict smoking. Epigenetics for biomarker development, I think the epigenetic clock is just the tip of the iceberg. This is because the effect of aging on the epigenome is pretty apparent, and we have the tools now to try to measure it. But I think as we keep getting better with our technology, we're going to develop more biomarkers for other things as well. And this is what we're trying to do, as I mentioned with my colleague, is trying to use epigenetics to target interventions and prevention, also to monitor whether interventions are working. This is an issue with the field of aging, right? Whenever they do studies, there's always a question, is like, well, how do you know it worked? Because you cannot follow people for decades to see who dies and who doesn't. those studies have not been performed. So they're always looking for biomarkers to see if an intervention actually worked. And I think this is a good start. And finally, I don't have to tell you this, right? Because a good start will last a lifetime. But I'm really interested in optimizing the prenatal environment. We study endocrine disruptors, things like metals, lead, and mercury as well. And I didn't present those data, but if you are interested, feel free to reach out to me. I would love to work on some of the fellows as well. With that, I just want to thank the students and postdocs. So I have a wonderful team. I was at UC Berkeley before, so a lot of these, the undergraduates and the PhD students remain there. And some of the postdocs are now here at Stanford as well. I've been fortunate to work with multiple bird cohorts. I presented data today from Gen3G from Canada, from Project Bio in Boston, and Chamacos here in the Salinas Valley, and lucky enough to also be funded by multiple NIH grants to do some of our work. And yeah, with that, I'll be happy to take any questions and hopefully have somewhat of a discussion with the remaining time.
All right, thank you, Andres. I think it's really obvious why Melissa and I were so excited about recruiting Andres here as really a joint partnership with epidemiology and pediatrics. During your original presentation and also during here, I was jotting down lots of things where, oh, there's a project with endocrinology, there's a project with GI, there's a project, but obviously, so much of what's already happening here. I'm thinking, for example, of SPHERE, this big U grant with Stanford Precision Health for Ethnic and Racial Equity, all kinds of operations to bring this into that work. Really, really exciting. I think one thing we were really excited about the possibility is our fellows, we now have mechanisms now for our fellows to get master's degrees in epidemiology. And I can imagine in every single one of our subspecialties, there's going to be some really, really exciting opportunities and sort of disease specific. You touched on allergy and immunology and asthma, and then also the prematurity. So just the possibilities are endless. So I hope all of you will think about the opportunities within your own divisions. And Lisa, obviously, the Office of Child Health Equity, I'm sure you can, the possibilities are limitless. Okay, so not surprisingly, we have some great questions to kick us off. All right, from Fernando Mendoza. If you haven't met Fernando, you need to meet Fernando. We will make that happen. Your data from Salinas for farm workers would suggest Latinos may have faster aging, yet Latinos overall have longer lifespan than other groups. Might you suggest that risk might change over time?
Yeah, I think this is definitely an interesting point about, I think this is the migrant paradox too, that some migrants fare pretty well when they come to the US. But it is, this population, specifically the Salinas population, these are all farm workers. And I think, it's pretty well characterized that they're at risk of exposure to pesticides, also disadvantaged conditions as well as an adverse environment. So I think we're dealing with a population that is definitely disadvantaged in many ways. And I think we're just capturing at least the farm work, the prenatal farm work piece was really interesting to me because I think it encompasses probably multiple domains that we're trying to disentangle, whether it's the pesticides, whether it's the social conditions, or whether it's other adverse conditions. Hopefully we'll get to that soon.
All right, there's lots of data in this lecture, so pardon me if I missed it, but what's the correlation between epigenetic age and age at death?
Yeah, that's an excellent question. Actually, epigenetic age acceleration as defined as the residual, so completely taking away the age, the effect of chronological age, that is more predictive of time to death than chronological age itself. So people are using it for risk prediction for that reason for insurance companies, as I mentioned. But yes, it is a stronger predictor of time to death compared to chronological age itself, which is one of the strongest predictors of time to death as well.
All right, there's an abbreviation in here that I don't know. You know what that is?
Oh yeah, the mother gene.
Okay, so can you comment on role of MTHFR on human health?
Mm-hmm. So I think they're talking about methyltransferases, perhaps. Perhaps I can clarify, but I think it's methyltransferases as well. And I think this is something that we're studying methyl donors like B12 and folate, not only for the prenatal period, but there's a lot of interest in trying to optimize the epigenome. Recently, there have been new developments in trying to program the epigenome using CRISPR and Cas9, the same way people are editing the genome, with the hopes that there's less off-target effects, that you're not truly altering the genome, you're just altering the epigenome. And B12 or other metal donor supplementations might be able to offset the off-target effects epigenetic editing.
Lisa,
Hi,
from
Lisa
Office of Child
Chamberlain.
Health Equity. Yeah.
Super exciting and so wonderful to have you here. One of the issues around public health is we have a hard time with precision, right? Some people can smoke their whole lives and do fine, or eat bacon every day and do fine, others not so much. How close are we to being able to start to say, and potentially even use in a pediatric practice, looking at kids and saying, you would be more resilient around X, or you would be more resilient around Y?
Yeah, I'm not sure how close we are to that.
Or what would be the area where we might start, like with asthma or some specific conditions that have high prevalence?
Yeah, and I think this is where the biomarker piece might help us, right? So let's say epigenetic agent makes it into the clinic one day, and that may become standard part of a panel. There's no reason why it shouldn't, and the costs are coming down and down as we go on. I think that can be an opportunity to try to understand if someone is biologically older, understand why. And this is what we're trying to do the research to characterize this. I think the reason why I think the epigenome is promising is because it's somewhat malleable. So when people stop smoking, actually the changes start reverting in a systematic fashion to the point that you know how long they quit, you know how long ago they quit, basically. So I think that's really promising to have a biomarker that is somewhat liable that way, that you can actually see time since quitting or time since they initiated smoking as well. So to monitor interventions, to monitor treatments perhaps as well, like with the asthma example that I showed you, you know, are the treatments working in the short term as well? But yeah, I think that's a great question.
Yeah, well, that's a great point, because the next question is, can epigenetic aging be reversed? Is that very powerful?
Yeah, well, I don't know if how many of you have kept up with this, but there was they founded Altos Lab, which is basically this research enterprise to study aging. And part of it, I think Jeff Bezos gave up the first round of funding in the order of billions of dollars. And that's what they're trying to do. They're trying to understand what can reverse genetic aging. There was a good example. There was a paper only in 10 men, middle-aged men, and they used human growth hormone, and they were able to reverse epigenetic aging. I'm not suggesting that anyone should be using human growth hormone, but they were able to at least see an effect, I think, with 6 months of treatment. Up to a year later, they reversed the clock for about 2 to 3 years. Whether, you know, I don't think the epigenetic agent should be the holy grail that we're trying to pursue in terms of modifying aging, but I think it can be a useful biomarker. But yeah, that's a pharmacological example. And there's now trials, people looking at lifestyle interventions to see if that can, you know, exercise and diet could reverse epigenetic aging too.
Great. Do we have a question?
Hi, I'm Kylie. I'm one of the PEDS residents. I've done a little bit of work with one of the former residents here about positive deviance inquiry around school readiness and positive childhood experiences. And I know you touched a little bit about it at the end of your lecture, but can you talk a little bit more about looking at the correlation between like positive childhood experiences and epigenetics?
Yeah, no, that's a great question. I'm glad you bring it up. I know the ECHO consortium, the environmental determinants of child health, you know, they're pulling a lot of cohort studies from the US to try to look at this. That has become a huge focus on like positive child health experiences. We personally have not. You know, I feel responsible that I'm always bringing gloomy news in terms of chemical exposures and climate change. And I think this is an area where we can actually try to find positive experiences, as you mentioned, or things that we can actually intervene in the right direction. But yeah, I personally haven't. As I mentioned, the parenting intervention that I'm working with my colleague in Florida is one example. And we're seeing some evidence that the intervention group has the accelerated genetic aging compared to the standard of care that the psychologists usually provide to the families. So at least there's some potential evidence that might be working. Yeah.
Thank you.
All right, great. That seems like a good note to end on. So with that, oh, did I miss anyone? Nope, I think we're good. All right, well, thank you, everybody. It was a wonderful conversation. And welcome.